on it, so
Hello.
I'm not sure what to do with it.
How are you?
Fine, how are you?
All right. Hello, Susan.
Hi,
Well,
and ask me how I am, and I'll go, too busy. I am too busy.
yeah. So, yeah, Dick will be about 15 minutes late. I got an email. I don't know who else will show up today. So at the end of last meeting, Susan was showing her presentation, and the last slide had to do with packings of spheres, I think, or it was a tensegrity thing, but packings.
Yes, people have made models, tensegrity models with packing.
Okay.
So I was trying to find that, and I found a GitHub. Let's see. Let me find that. Yeah, GitHub page there. Okay. Would you like to see the GitHub?
Yeah, it'd be good.
Okay. Try to share my screen. This is a little different. Window. Okay, there it is. Can you see that?
No.
You can't see my.
I can see the window for Jitsi, but not you shared the tab instead of the whole screen, I think.
Not again. I am having some trouble with that. Let's see. Oh, entire screen, that might work better.
Yeah.
Okay. Entire screen.
All right.
Okay.
There we go.
So can you see that now?
Yes.
Okay. Well, this is the GitHub thing that I found. So I'm not sure. Are they doing some of this in MATLAB? Anyway, there's code.
Great.
Code for this.
And they have a finite element model. Looks like.
Yeah, so they're finite element models, and it can do just tensegrity structures, and then it can do form finding, and then finite element analysis. So this is pretty good. I should try this or show this as an alternative to what I'm doing. So it says this is, where did it say? Yeah, it says MATLAB function. So there's some MATLAB, and I have MATLAB. So it would be, I've already told the professors, they said, no, don't do that. You're being, you're going off track. No, I'm not going off track. You have to have some sort of, you have to do a thorough job of pointing out what other people have done as well. So,
right.
So this is fine. This is some MATLAB code, and it's on GitHub, and I might actually be able to use this.
Yeah. So go down to the next page.
Some of this, sorry, some of this is looking like my exterior cells, epithelial cells. Right. Anyway.
So form finding is like finding the different shapes that they make, like the different topologies.
Well, it's a different topology for wherever you, whatever you start off with. I think this could be form finding.
Yeah.
If you start off with this. structure and then it'll form find until it's more round. No, this is 6 strut and this is 12 strut. I don't know. It finds the most stable structure. I've seen it with various programs. Like I have a couple hundred papers on this stuff.
Right, yeah.
And they're always going into form finding, and I still think if you leave a single cell out, it's going to become more round. Usually, unless it has attachments.
What do you mean a single
That might
cell? A single cell
I know.
or a single?
Single cells are different than cells in tissue.
Oh, okay. Oh, if you have a single cell versus the tissue, then
Yeah.
it becomes more
So
round.
anyways, yeah, so I added the GitHub to this, and I didn't find much more, but I did find this with Actin networks. So I put some Actin network stuff in here.
Okay.
So you have disordered, ordered, and disordered bundles. And this is what they'll do for you. If you have ordered bundles, they tend to would close. If they're a ring, they'll close if they're an ordered bundle. And a much better than a disordered networks. Well, they sort of do it. So looking at this might be wound healing that Dick was talking about. I should probably be showing him this, but anyway, this kind of looks like what happens in wound healing in tissue.
So it's getting smaller. It's contracting back down.
Yeah, it contracts down.
Okay. And then the disordered bundles are, I don't see the difference between the disordered and
disordered networks sort of close, but not completely.
Oh, okay.
And the disordered bundles don't seem to do anything.
Oh, okay. Huh.
And if you go back to this is the difference. So these are disordered branch networks, ordered bundles, and this I think this is like myosin in between here.
Yeah.
And then you get bundles that are disordered and they don't tend to do anything.
Yeah. So just the configuration. Yeah.
The configuration of the actin is important.
Yeah.
So this is what I know from the course on Coursera, cytoskeleton course in Coursera, is that if you have if cells under low force, they tend to do this. And as the force increases, the angle between the actin changes, and it also tends to grow a whole bunch of actin bundles at the surface if you have it under high force. And if this is an adaptation, so if you leave the force on it, this is what happens, and the tissue relaxes, but it's changed.
Okay.
And it has more actin at the surface.
Okay.
So, yeah, relaxation is an important thing. Like if you apply a force and then leave it there, the tissue will adapt to that, and it's important for just the biomechanics of cells. especially during development, because you're pulling and pushing on the tissue, or it's pulling and pushing on itself.
Right.
And this is, yeah, so this is also important in bone development. If you don't have something like this occurring in bone, you don't get proper bone development. So it's sort of an interesting thing that I learned at the March meeting.
Okay.
I'm going to the APS March meeting.
Yeah.
Again this year. Anyway, so if you have a low force, you get this type of configuration. And then you get high load, and you get more ends created, and you get this more of a 90-degree branching. So that goes with what Dick used as sort of the microtubule and acting together to stabilize the microtubule. These branching ends will stabilize the main branch of these microfilaments, or actin filaments, rather. Do you want to see that?
Yeah, it'd be good. Oh, you have a physical model. I see.
Well, I can't really do it by myself, but I'll just. You can see me? Oh, there. So if you take a very thin rod that will bend easily, and you take elastics. These can just be plain elastics. I've got these so that you can see them. And you pull on them like this. The rod won't bend as easily.
Okay.
Like you stabilize it by pulling on the sides.
Yeah.
So if you see something that's branching, that might be causing it to, well, it might be stabilizing the middle portion of it. I don't know. This is better done in the classroom.
Yeah.
Where you have everybody take a hold of an elastic and pull. And then you can see the difference. This becomes very stiff. Instead of bendable, it all of a sudden becomes stiff because you're pulling on the sides.
You mean the part between where you're pulling? So you're pulling at 2 points.
Yeah, this is the middle part, and it is flexible. But it won't be flexible if you pull on all of these elastics in different directions. You need to pull on them from different sides. Oops, I just lost a few on the floor. But anyway, yeah. So it's just how you stabilize a flagpole, for instance. You put guy wires down to stabilize it. So you're stabilizing a floppy thing like a microtubule or a microfilament bundle, or an actin filament bundle. It's floppy, and if you pull on it from the sides, then it tends to make it stiff in the middle.
Yeah, that looks great.
Anyway, maybe I need to do something about getting a little movie of it.
Yeah.
Or something.
Yeah, well, yeah, that would be, but the demo would, if you had like several people to pull on it.
Yeah, if you have other people to pull on the elastics, and like I said, they don't have to be fancy elastics, they just need to be some sort of elastics, like plain elastics.
Yeah.
So that's part of why it makes the cell surface stiff. You get all those branching actin bundles at the surface, and you push on your cell, and you get a stiff surface. Oh, and there I wanted to show you these. See? So I was showing the microtubule with the actin, or the actin with branches, these being elastics on a flexible, I don't know if you can see that. You can see me, right? This is with the elastics. These are fancy elastics.
Okay.
He knows he's the one that showed me this in the first place.
Oh, okay. Yeah, it does look like a dick thing. Yeah.
Anyways, so I'm saying that this is what's happening here with the microtubule, not microtubule, these actin filaments. And what happens is they do this. You add a force, and the angle between the actin filaments change, and then they create more of themselves and make the surface here stiff. And this is, I'm saying this is important in bone development, for instance.
Okay, Susan, what happens in microgravity?
The cell has a lot of trouble producing its cell cytoskeleton. At least at first, it has to adapt to microgravity, and it still has trouble. I think it takes more energy to create this structure. And the reason why is because I think cells use convection and little convection, and
okay, so what we need is micro robots to go inside cells and cause convection.
Well, we're better off than plants, because plants just sit there. Like, at least we move, so we're causing some convection to occur in our system.
Okay.
Oh well, and here's I was just showing them this slide. So these are actin rings that they've made, and if you have disordered networks, they sort of close in on themselves. But if you have ordered bundles, you get a closure here. So this is kind of like wound healing, I guess. And then if you have disordered bundles, nothing happens.
Okay. I recall for the axolotls when they did this, the radius went down by about a factor of 10. It's more than this does.
Okay. Maybe they have very ordered bundles.
Maybe.
I don't know. I don't. But this is after 15 minutes, so maybe if they'd left this one for a few more minutes, it would have done more. Or maybe it didn't matter.
I think the axolotls do it in about 3 minutes.
Oh, okay. There must be some myosin. I'm going to say there's myosin associated with the actin, because there always is. Only in this experiment they didn't put it in there. Although it looks like they did, because this looks like myosin in here.
Oh.
So I'm not sure.
Small oversight.
Yeah, I don't know if it's an actin myosin thing, or if these are just simply myosin that are ordered. Okay, I need to read this paper a bit more, or read it over again. It's called reading it again. But yeah, if you had some more myosin in here, I think you'd get contraction happening in a hurry. Get some myosin and some actin and throw some calcium ions at it. You'll get contraction. There you go. So that's what I added to this. And I found GitHub with tensegrity models on it for my essay on tensegrity and modeling, which I have to do. And then I also found some tables where they'd ruined the cytoskeleton and then looked at its reaction force due to compression and shearing, but they didn't do tension. So I say their experiment is incomplete. I need to do tension. Anyway, that's what I added to the slide presentation that I did last week.
That's great. Yeah, thank you. Yeah, that was a great presentation. Okay. I assume you'll be adding to it on a regular basis.
Yeah, it's already fairly long, and I suspect I'll make it even longer, and then I'll just take it and make it into an essay. So
I budget. I used to have lectures that were almost 3 hours.
Oh no! Well, I think I'll send this to Stephen Levin if he said he'd like to see it. So
yeah,
since he went to the bother of helping us out with this, so yeah.
Yeah, you might add your observations on microgravity.
Oh, okay. Yeah, it might be interesting. Okay, well, I could put there that.
After all, people want to spend 6 months going to Mars. They may end up getting there and being mush.
Yes. Okay. There you go.
Oh, okay.
All right, I'll add something to it. Okay, well that's the end of that. So
okay.
I'll stop sharing.
You can get some NASA money for the simulation.
Oh, yeah. No, they might be studying biology now.
Send yourself to in orbit for 6 months and see if you survive.
Oh, okay. They want to send me to the moon. That's the next thing. They want to send people to the moon. Yeah, I don't know, that little bit of gravity, do you think it would help? Or is it still going to cause...
Not if you don't bring air.
It'll cause dysmorphologies.
So let me share a few updates with you and some features that I have. So the first update is on the DevoLearn and DevoGraph repository. And so this is a kind of an important thing. We had Sushmanth Reddy, who filed an issue in the DevoGraph repository in the DevoLearn organization. So we're at the DevoLearn organization, we're in DevoGraph, and he found a pretty serious issue with respect to dependencies. So we have some dependencies issues in the DevoGraph stage 1 pipeline. This goes back to like last summer's GSOC and some of the things we did over the fall, where we were modifying the stage 1 pipeline. This is the pipeline for the DevoGraph platform. So stage 1 is where you get segmentation of images, and it ties into DevoLearn as well. So Sushmanth made an observation here. He created issue number 1. So it's open if people want to solve it or if people want to address it. You can leave a comment down at the bottom. So Sushmanth says, there is a problem with DevoGraph stage 1 pipeline while extracting centroids of cells from DevoGraph stage 1 pipeline. There is a dependency issue that DevoLearn is encountering. There are a lot of deprecated libraries that are being used for DevoLearn. These are creating a problem when we run the Colab notebook. So if you run the Colab notebook for DevoLearn, there's a problem with dependencies that you hit, especially also if you're integrating that with DevoGraph. And the libraries for these should be updated in DevoLearn. There's a requirements.txt file for a smooth movement stage 1 pipeline of DevoGraph. So he's documented the following errors. There are a number of, so scikit is deprecated, NumPy is deprecated, pandas is deprecated. I'm not really sure, there are probably errors in the code that need to be resolved, but it could also be that the way that these packages are referenced is out of date. So if someone wants to take up this issue, you're more than welcome to do it. Sushmanth will probably work on it. He's consulting with Mayuk and also with Jiahong, who's the sort of the ringleader of the DevoGraph group. So again, you can jump in. This is open source. You can take a look and see if you can fix it. Open up an issue or a pull request, and we'll get to it. I think DevoLearn doesn't have anything at the moment, nothing open. We actually have 3 pull requests open and 8 issues. So if you're interested in looking over DevoLearn, this is going to be for the Summer of Code project this year, key to making DevoGraph run. And so we're trying to get all these bugs worked out by the application period for Summer of Code. But this is also something if people want to, propose a project, part of that project might be fixing some of these bugs and making sure everything is up to speed, because that's one part of open source that's very important, is being able to fix bugs and fix dependencies issues when needed. So this is a good thing to kind of engage with before the Summer of Code, but also, even before the Summer of Code, if you want to make a nice open source contribution, this would be the place to do it. And so you could go to the DevoLearn organization at github.com/DevoLearn, either go into the DevoLearn repository or the DevoGraph repository and check out the open issues with respect to these problems. Okay, now I wanted to go over a few features that I had, and these are things that I found over the course of the week. So one of the things we'll be talking about in this meeting are the origin of life and lipid membranes. And so there are 2 features I have for that. The first one is from Cybertron blog, or Cybertron Asks blog. And the tagline of this blog is what, why, and how do we know? And so this is, I think they're taking a free energy principle approach to lipid membranes. The title of this blog is Markov Blankets versus Lipid Membranes. So they start by talking about Nick Lane, who is an excellent popular science writer on biochemistry and some other things. He has a book out now called Transformer. It's not the machine learning type of transformer. It's actually transforming chemistry into biochemistry at the sort of the origins of life. And so it's a really interesting book, very wonderful set of references, a nice annotated bibliography at the end of the book. And it really frames some of these issues in the field nicely. So watching this wonderful Krebs cycle RI lecture by Nick Lane, this is the lecture here. I'm not familiar with the RI series, but I just wanted to note 2 things for now. One, there is an obvious topological parallel between systems, Markov blankets, and mitochondria, lipid membranes. So there's a systems level construct of a Markov blanket, and this is something that the free energy principle relies upon to sort of build the boundaries around systems. So a Markov blanket is something you put over the states that you're interested in solving for, and the blanket can be approximated to give you a boundary of your system. Systems boundaries have always been a contentious issue in systems modeling. Finding out where that boundary is and giving a justification for it, not just in a definitional sense, but in a mathematical sense, has always been very difficult. So Markov blankets give you a tool to do that. And then lipid membranes are, of course, these membranes that separate the inside of a cell or the inside of a vesicle from the external environment. Its permeability is variable. We'll talk about that later in the meeting. And you have this, so it's built out of lipids, and it's selective in what it lets into the cell or the vesicle and what it excludes. And so this is important for compartmentalizing biochemical processes or other types of things that we might be interested in. So it provides organization of life. So this actually gives you abstractions at 2 different levels of systems: the information processing aspect and the biochemical processing. So lipid membranes allow you to think about systems boundaries at the biochemical level, and Markov blankets allow you to think about this at the information level. But also Markov blankets give you a route to thinking about this in terms of thermodynamics. So it links them together nicely. So that was the first parallel. And two, early on, Nick Lane makes quite a few remarks against the informational computational view. So this is kind of what we just laid out in the first point. But, some people don't view that as something that the way you should view nature. So, some people don't think you should view nature from a computational point of view, that it's not really relevant to what's going on. But, this is something that we use as a way to get a sort of a quantitative handle on things. So he makes quite a few remarks against this point of view. It highlights the information element when we get to the nucleotide process. So fascinating for the metaphysical aspects of which came first, which is, of course, what his talk is about. So this is in the book Transformer. He focuses on the Krebs cycle and the reversibility of the Krebs cycle and how that contributes to sort of the origins of life and the early evolution of life. So the primacy and universality of Krebs cycle in anything we recognize as biological life, that's one of his points in the book. From the simplest physiochemical precursors to the most complex multi-celled creatures. So the Krebs cycle is proposed to be sort of one of the things that defines LUCA, which is the last universal common ancestor. And then from there, you see it in every form of life. So this is something that we can focus on if we want to think about what does all life have in common, and it's probably a Krebs cycle of some type. Or there are other features like maybe a systems boundary, like a vesicle, or some other more abstract boundary. And so these are the kinds of things we have to think about in terms of the last universal common ancestor. He also thinks about the inevitability aspect, that once the simplest chemistry exists, evolution in the complex follows. So in other words, you have this simple chemistry, and it's inevitable that life will evolve from that, and that complexity will emerge from that. And I think that's kind of an interesting point, because people tend to always throw around this term that life is sort of rare, that you don't see it everywhere. And yet maybe you do. Maybe life does emerge a lot more than we think. Maybe there are certain constraints that have to be in place that once you get life and it starts to sort of take different niches on a planet or in a set of ecosystems, that you can't have life emerge again. But we don't really know that. We don't really know, say, what's going on other planets, or we don't even know what's going on in the extreme environments on this planet very well. So this is interesting that you have the simple chemistry, and that actually life might be quite common with respect to that chemistry, that it bootstraps a lot of living processes by default. So this is just an interesting talk. This blog post kind of doesn't talk about it that much more, but it does get into some of these issues that we're interested in the meeting. And so this actually connects with like this sort of non-neuronal cognition that we talk about in the group a lot. So this set of tweets from Ian Glendening is in response to Wiring the Brain, who's actually a very good resource for systems neuroscience on Twitter. And he talks about really is a wonderful lecture, partly because he's so good at it, but because of the sheer scope of levels of existence involved, from origins of life and consciousness via cancer, aging, and diabetes. So he links the Krebs cycle to all these diseases, which people are more interested in than the origin of life, to the whole Earth itself as a self-regulating organism. Mind-boggling. And Mark Solms, right at the end, see video, the idea of affect feeling is an electrochemical report on the categorical qualitative state of the cell. So this is interesting because you have this idea that cells have feelings, but not the feelings that we have in our brains. They're different types of behavioral signals or signals of a state of being. So affect and feeling might be, if the cell is sick, it has certain gene expression profiles which you could activate it, and that results in an electrochemical signal that you can then measure, and it's very different from the cell in its default state. Or, as the cell ages, we know that there are different markers that get upregulated and downregulated, and again, these move into some of these electrochemical assays that you can do, and it tells you something about the state of that cell. That's a different category of being than, say, like a normal state. In genetics, we often talk about a wild type versus a treatment or a mutation. So the wild type is basically the normal state of the cell or of the specimen. And so in a cell line, for example, you could have a wild type cell line. It doesn't have any of the mutations that we might knock out a gene or we might add in a gene. And it's basically what you would find in nature. And so we think about a lot of biological experiments in terms of that wild type versus treatment or versus mutant or whatever. This suggests, of course, that you can have these states that deviate from the normal, from the wild type state. And maybe that wild type state is a little bit overstated. Maybe there are a lot of different qualitative states to the cell that transcend the wild type idea or the control or, a normal state. So I think it's really interesting stuff. There's a lot of stuff at the intersection of course, origin of life in this work, but also of non-normal cognition in this work. So Jeremy England, which we talked about a few meetings ago with his new paper, he asked a question in this talk midway through the Q&A. He talked about the last universal common ancestor of both archaebacteria and bacteria, mitochondria and the eukaryote cells. So all these different, kingdoms, their last universal common ancestor. None unlikely coincidence. Everything to sew with structure, not info. Yes, yes, yes. Systems architecture level info, not individual bits. That was a little bit, obviously it was paraphrased, but the idea being is that you can look at some of these things as structure instead of information, or like this computational view of information where you're looking for bits. You're actually looking for structure, and it gives you systems level information that we can't. So it's just informational bits might be viewed as a sort of reductionist quality, whereas the systems level architecture might be viewed as this systems level or emergent quality. And so this ties back to some of the qualitative states that might exist in cells. So I think this is good. And if you want to know more about the Markov blanket, Carl Friston has worked on this for a while, and this is a part of the free energy principle. So this is there's citations on this out in the world, and there are some other notes in here. So yeah, this is a little bit beyond what we were talking about, but so take a look at this blog post if you want to know more, and I'd be interested to hear your thoughts. The other feature I have for you today is this Origin of Life Digest, and this is from Alexandre Champagne-Rouel, who's another blogger. He has this Origin of Life Digest, so it's a digest for every week. He puts out a digest of preprints and papers in the field of the origin of life. So this is this last week, January 30th. We have some papers in the artificial life field, from astrobiology, biochemistry, biology, microbiology, paleontology, planetary science, statistical physics. And so it covers a lot of different fields. There are all these different research topics that they explore, and they kind of come from different angles. So it's nice to have all these papers in one place. So I'm not going to get into any of these papers, but I wanted people to know that this existed. I don't know how long he's been doing this for. Looks like this goes back, last year, obviously. Wasn't that long ago, but there are a bunch of digests that you can go back and look through if you're interested. So in keeping with today's meeting theme, I'd like to talk about some papers from the origins of life or early life. And so we have these papers on what I call ancient systems. So these are kind of going over some of these issues, not at the origins of life, but in early life. We've talked in past meetings about the boring billion, before cells were really, or life was really exposed to a lot of oxygen. And so it was very simple, and it kept it very, unicellular, and there weren't a lot of innovations. And as soon as you had an increase in oxygen on the planet, we got a lot of different types of life and different types of complexity that was innovated by life. So the first article is this Quanta article. A billion years before sex, ancient cells were equipped for it. And this is based on molecular work that's zeroing in on the origins of sexual reproduction. The protein tools for cell mergers seem to have long predated sex. So what were they doing? And so this goes through some of this. In researchers' quest for the origins of the ability of gametes to fuse sexually, they were looking at related genes in primitive archaea bacteria and viruses. So that's what this article was about. I talk about sort of the genes that you find, common genes in organisms that we don't think of as being sexual, but they're nevertheless there. And they manage this reproductive ability. Most complex organisms engage in a strange bit of genomic math at some point in their lives. To multiply, they subtract and then add. That is, to reproduce through the process of meiosis, they create specialized cells, or gametes, with half the usual number of chromosomes. They then merge pairs of these gametes to create new individuals with a full unique genome. Sexual reproduction is nearly ubiquitous among eukaryotes. Organisms from kelp to koalas that have a membrane, bound nucleus, and organelles. So the cell, of course, has a membrane. The nucleus is membrane bound within the nucleus, and we have organelles inside, such as mitochondria or chlorophyll, that perform specific functions. This is chlorophyll, of course, being in plants, mitochondria being in animals and plants. We animals merge a sperm and an egg. Mushrooms sprout from the underground collision of fungal threads, pollen scent tubes racing through floral tissues that join ovules, creating fruit and seeds when they meet. Yet fusing cells like this runs contrary to normal cellular life. So these cells, the origin of eukaryotic cells was that they fused together from multiple organelles that were floating around in the early environment. And so this is not typical of a lot of early cells and just sort of the origins of cells. Yet fusing cells like this runs contrary to the normal cellular life cycle. Cells divide in two throughout mitosis to produce asexually, but otherwise they mostly guard against major disruptions of their integrity, which could snuff out their lineage. So you see this after the end of the boring billion, you see a lot more diversity in terms of cells living in groups and being able to do things like combine different organelles into a single cell. You see a lot more complexity, and it's based on this idea that it's not a typical thing that cells do. So why this hazardous arrangement caught in an evolution has been the subject of intense study. It's generally presumed that sexual recombination helps to keep a species' genome healthy and diverse, but researchers have also been piecing together molecular clues about how it evolved. So in the asexual organisms, you typically get you lack the ability to generate genetic diversity. You get mutations, but that's often not enough to keep up. You need to have recombination. You need to have mechanisms for keeping diversity flowing in a population or in a genome itself. Throughout nature, gamete cells like eggs and sperm fuse to create new genetic individuals, or zygotes. But outside of sex, these kinds of fusion events are far from routine for eukaryotes. So you don't really see that everywhere. You just see it in the germline and sexual reproduction. And so this is a very similar thing to what happened in basal eukaryotes, where you had this merger of organelles into a cell. You also have this merger of fusion of eggs and sperm to create a new type of cell, at least from the standpoint of what came before it in that interaction. So there's this paper on the bioRxiv that suggests some cells have the power to fuse their membranes far back in their evolutionary history. The molecular machinery that makes this part of the sexual reproduction act possible may have existed more than 2 billion years ago in the simple prokaryotic cells of archaea, perhaps as much as 1 billion years before eukaryotes and sex evolved. But new findings also hinted at an explanation for why this kind of cell fusion didn't appear earlier in life's history. So this is where you get these fusion proteins, a family of them, and they evolved fairly early on. So throughout the family tree of eukaryotes, the same protein enables the cell's membranes and nuclear membranes of gametes to fuse, and this is HAP2. Because it's shared so universally among eukaryotes, the common ancestor must have had the same mechanism. You'll see this paper, Archaeal Origins of Gamete Fusion. This is authors on this paper. And so if we go to the bioarchives paper, we'll see that there's this title was Archaeal Origins of Gamete Fusion. So it talks about gamete fusion and its origins as a family of genes. So here, using bioinformatics, we identified archaeal genes encoding candidates of fusexins, a family of fusogenes mediating somatic and gametic fusion in multiple eukaryotic lineages. So they're able to assay some of the aspects of this these candidate genes, and they were able to look at like the proteins that it produced, and they were able to look at how these things fuse mammalian cells. And there's a conserved fusion loop, so you see this throughout eukaryotes. And genome content analysis reveals that archaeal fusexins genes are within the integrated mobile elements. So these are your integrated mobile elements, transposons that allow have other functional significance. Finally, evolutionary analysis places these archaeal fusogenes as the founders of the fusexin superfamily. Based on these findings, we propose a new hypothesis on the origins of eukaryotic sex, where an archaeal fusexin, originally used by selfish elements for horizontal transmission, was repurposed to enable gamete fusion. So there are these horizontal gene transfer elements in archaeobacteria and bacteria that they're selfish elements, so they sort of transfer within the cell, and they're able to transfer to different cells and fuse together. And this is something that then was repurposed for sex. And so now, if we go back to our article, you see here's an example of the assays that they did here. These are the HAP2 proteins, like the one modeled here, enables membranes of gametes to fuse. The structure of HAP2 links it to a superfamily of fusexin proteins with similar functions in diverse organisms, and this includes viruses. So in the paper, they were talking about the function of this in archaebacteria, but it also functions in a very different way, but with a similar gene family that produces a similar protein in eukaryotes, and even in viruses, it has a function of its own. So this is, they talk more about this. They talk about functions before fusion. But in some way, it's also a bit predictable, because basically the whole machinery for meiosis has an archaeal origin, which is interesting. We don't really think of meiosis as having an archaeal origin, but here using molecular data, that's what they found. And so they have this figure how two cells become one. This shows the fusion of gametes, and it shows that there's a half set of chromosomes in each gamete, and they combine to make a full set of chromosomes for what will become a somatic cell. And so you have this, basically this is how fusion proteins bring cells together. So you're basically taking two cells and you're transforming it into a new type of cell. And so this sort of thing happened at the origin of eukaryotes. It happens in sex all the time. It doesn't happen everywhere in the body. And it has an archaeal origin, and it even has some parallels with what happens in viruses. So how fusion proteins bring cells together. Gametes can be combined because of a special class of fusion proteins, fusexins, on their membranes. So these are the fusexins on the cell membranes. They bring the cells together. They open up this patch here that allows things to transfer as a bridge between two cells. And then as the membranes open up, then they share what's inside the membranes. And that's how you get your cell fusion. So it's really interesting stuff, especially in light of some of the early work on vesicles and membranes, which I'll talk about later in the meeting. There's this other paper on billion-year-old multicellularity. So this is from Current Biology, a possible billion-year-old holozoan with differentiated multicellularity. So this is the graphical abstract where you have a protist, a billion-year-old protist. This is a new genus Bicillium, which demonstrates cell-cell adhesion. So we have the naked stage here. There's differentiation and cell elongation in this colony of cells. In the naked stage, they seem to be undifferentiated with respect to size and shape. But in this cell elongation stage, you get these elongated cells, and then you get differential adhesion and cell migration within this mass, and you end up with a cyst stage. So this is the life cycle morphogenesis of this billion-year-old microfossil, which they found, that actually may be key to the origins of embryonic development. So we've talked about the Doshanto findings from China, where they found some very old embryos that they think are embryos. They don't actually know that, but they look like modern embryos. And it's like the first example in the fossil record of this. This is actually pushing this back a bit more. This isn't necessarily an embryo again, but it has a lot of the properties of embryos, and it shows a lot of the sort of the things going on in embryogenesis. So the highlights of this paper are that the multicellular microfossil Bicellum brachyeri possesses two distinct cell types. So you can see the cell types in here differentiating at this stage, these elongated cells and these more rounded cells. And then these push out to the outside and they become what looks like this ring around. This almost looks like an inner cell mass with an outer shell. So this is like the cyst stage. So this is why people say this looks like an embryo. Now it may not be an embryo, but it's doing these same sorts of things that an embryo does. 3D preservation and phosphate preserve different life cycle stages. Differential adhesion may have contributed to cell segregation during morphogenesis. And this billion-year-old freshwater protist shows evidence of holozoan affinity. So it's affine to things that you find today. It's related to things you find today. So this is from the Northwest Scottish Highlands. This is these are microfossils from a non-marine setting about 1 billion years ago. Phosphate nodules from the Diabog Formation at Loch Torridon preserve microorganisms with cellular level fidelity. So this is a very nice preservation at the cellular level. So they're able to reconstruct developmental stages of this organism, Bicellum brachyeri. The mature form of Bicellum consists of a solid spherical ball of tightly packed cells, a stereoblast. It's what we call a stereoblast when we look under the microscope. In isodiametric cells enclosed in a monolayer of elongated sausage-shaped cells. However, two populations of naked stereoblasts shows mixed cell shapes. So this is where we're looking at two populations of these cell types, or these cell formations show mixed cell shapes, which we infer to indicate incipient development of elongated cells that were migrating to the periphery of the cell mass. These simple morphogenetic movements could be explained by differential cell-cell adhesion. So as cells move around, they adhere to one another, they don't adhere, and that produces the effects of sort of sorting as they migrate. In fact, the basic morphology of Bicellum is topologically similar to that of experimentally produced cell masses that were shown to spontaneously segregate into two distinct domains based on differential cadherin-based cell adhesion. The lack of rigid cell walls in the stereoblast renders an algal affinity for Bicellum unlikely, since its overall morphology is more consistent with a holozoan origin. Unicellular holozoans are known today to form multicellular stages within complex life cycles. So the occurrence of such simple levels of transient multicellularity seen here is consistent with a holozoan affinity. Regardless of precise phylogenetic placement, which means where is it in the tree of life. These fossils demonstrate simple cell differentiation and morphogenetic processes similar to those seen in metazoans today. So this is similar to animals. This is their site that they've gotten these fossils from. Then these are some of the pictures of this Bicellum brachiaria in its mature form. So it has this shape that we see at the sort of the third stage of that graphical abstract. This is the mature stage of Bicellum. And then we see variations on this. We also see in this one, this is the distributed form here. These are the naked stereoblasts. So this is sort of like the first two stages of that graphical abstract. You see how you have like the parts of the stereoblasts that are single, undifferentiated looking cells. They all look basically the same. There isn't that much organization around the edges. And then you move into these other micrographs C and D, where you get these elongated cells and they're starting to migrate around this mass. And then in E and F, you start to get these zones where you get a more defined edge, more defined edges. You start to see the elongated cells move out towards the edges, and then you start to see the structure in the mature organism. So this is a good paper. I think that's good evidence for billion-year-old multicellularity and also of very early embryos. To wrap up here, we're going to talk about the origins of arthropod brains. This paper, I think it's from Science. Yes, and this is from arthropod evolution, the lower Cambrian, Lobopodian, Cardiodictyon resolves the origin of eutheropod brains. So this is sort of the origin of brains in arthropods. So for more than a century, the origin and evolution of the arthropod head and brain have eluded a unifying rationale, reconciling divergent morphologies and phylogenetic relationships. That's a long way of saying they really don't know how to figure out which parts of how did the brain evolve from a common precursor, or how did it evolve from a very simple form. Sometimes it's easy to reconstruct these relationships, and sometimes it's not. In this case, it's not. Here, clarification is provided by the fossilized nervous system of the lower Cambrian Lobopodian Cardiodictyon catenulum, which reveals an unsegmented head and brain, comprising three cephalic domains, distinct from their metameric ventral nervous system, serving its appendicular trunk. Each domain aligns with one of the three components of the foregut and with a pair of head appendages. Morphological correspondences with the stem group arthropods, which are the origin of arthropods, and alignments of homologous gene expression patterns with those of extant paranthropods demonstrate that cephalic domains of this organism predate the evolution of your euarthropod head, which is euarthropod is the larger group of arthropods. It corresponds in neuromeres, defining brains of living some other, like the mandibulates and the Cheirocerites. So I'm not familiar with the former, but the other is organisms with mandibles. So they have this, they talk about the origins of the brain. They talk about some of the early parts of the brain. So this is, so we have this, they show the segments of this arthropod. So again, this is fossil evidence. And you have the origins of the arthropod and its segmentation. You have these cells here in B, which are, you have these shield-like sclerites that are bilateral punctate thickenings. So this is something that defines each segment. And you have these legs that come out of the sclerite side braces, which are these. So each of these segments form this little thing here in the middle, and then these legs. And this is in each one of these segments, you have this sort of, it's like something that allows the organism to keep its structure. You have a gut here that runs down the middle of the body, and then you have the brain at the anterior end of the organism. with different aspects of it. So this J shows this organismal configuration. In F, G, H, and I, it all shows the sort of the anterior end of the brain. So you have some decent preservation for some of these structures in the early arthropod brain, and you can see it in this diagram. Correspondence of these different structures. And so moving on through the paper, we have the segmented nervous system and segmental brain. So all this is just showing evidence that there's segmentation in the body, there's segmentation in the larger nervous system, but the brain itself in the anterior end is asegmented. And so this shows more evidence of this. And then this shows the correspondence of nervous systems. So this shows the cerebral domains and the corresponding neural mirrors in the fossil to living organisms aligned. So these are your different comparators. You have gene expression spatially restricted here. You see these lines, they show these different genes that are expressed in different parts of the organism. So, invertebrates are a very good example, or insects are very good examples of restricted gene expression. So you get gene expression in different segments of the organism. So you see, for example, the Hox genes are expressed at different parts of the organism's length. And then you get these other genes that are expressed more in the brain. So PAX6 is expressed throughout. 6-3 is expressed sort of in the anterior end. And you see this across different taxa. And you see this, how this can resolve in the formation of an anterior end that's not segmented versus a body that is segmented, or at least a non-brain body or non-brain part of the nervous system that is part of a segmented phenotype. And so this just shows some of these details of the nervous system. It's showing that you have this anterior end where there's a non-segmented brain, that there are different ways that this can be done. And then that's that paper. So this shows, again, fossil evidence of early brains. And we've also talked about the origins of multicellularity and functions such as sexual reproduction and the integration of cells and other types of structures and how that occurs. And its origins that predate metazoans. Thank you. Hope you learned something. Probably, yeah.
Yeah, you better bring air and lettuce. Lettuce is very expensive on the moon.
No, yeah.
Oh, okay.
Okay.
Yeah.
Hey, Bradley, you want me to go over that proof I sent?
Yeah. Let me pull it up.
Okay, this is a proof. That's why it's all marked.
Oh, okay. Yeah.
Okay. Okay. Now. Okay, let me skim through here. Okay, so the basic problem is that membranes seem to have been half as thick when the first membranes were formed.
All right.
Okay. They were made of amphiphiles, which means a molecule that's hydrophilic along its length and hydrophobic along its length and hydrophilic at least one end or both ends. Okay, so it's got a charged group at least one end. Okay, now let's see if I can turn this. Turn it. Oh, come on. Okay. You. Now, just rotate on this program.
Okay.
Rotate things there.
Yeah.
Okay, I'll just rotate. Now,
Should be okay.
right, there we go. There we go. Okay. Okay, now. there's a lot of speculation about which amino acids were the first ones on Earth. Okay, so one thing we did is compile a list of the 20 amino acids. We'll take this. Oh, boy. Oh, okay. Okay, and these are the number of references. So what we found is if you take a given amino acid, this one, for example, 8 papers said it was first here.
Right.
Cysteine, none. Glycine, 15. So we're looking at a consensus. And basically, what we concluded is that there is no consensus. If you look at the number of references.
Right.
Okay? Which amino acid came first. Okay?
Yeah.
So what we did is we turned to, let's see. Okay, I want to show you an interesting thing here. Let's see. If you go to, there we go. Okay. Okay. If you look at the lipid chains, they have a curious property in that they're melting. They would stick together and form a two-dimensional fluid. Okay, but their melting temperature saturates curiously very close to the boiling point of water. Versus n is the length of the chain. Okay, so this indicates that there's some phenomenon which might limit how long an amphiphile can be in a membrane. Okay. Here. Okay, here I did the number of stereoisomers versus the log of the length. A stereoisomer is a molecule with the same number of carbon atoms or same number of atoms, but in a different configuration. And if you look at this table, this is N, the number of structural isomers, the number of stereoisomers. So stereoisomers would include those that are mirror symmetric. And look how this table goes up.
Yeah. Wow.
Okay.
Yeah.
Okay. Yeah, here's even more. Okay. So that's a combinatoric calculation, but
Right.
it's sort of the number of trees you can make for that number of molecules, for that number of atoms. Okay.
All right.
So you can see that number goes up very fast.
Right. Right.
Okay. Now, if you look down here, it's not absurd when you're down around 16, but it gets pretty big later on, which means that if these things are made randomly, the ones that are straight will be a smaller and smaller proportion.
Okay.
Okay, so let's see what else we got here. Okay, so these are classes of amino acids, how they're classified. Okay. Table of that. Now let's see. Okay, now an antimetric excess is an interesting concept. If you have a mixture of left and right-handed amino acids, mirror symmetric, it turns out that the proportion in different meteorites is not 50-50. Okay?
Yeah.
They have an excess, and these are the estimated excesses. There are various ways of calculating the excess, but basically, the closer you are to 0.5, the closer you are to what's called racemic, where you have an equal mixture. Okay? Okay, so you can see there's a bias already in meteorites towards amino acids of the types that we have in living things now. Okay?
Yeah.
And where this comes from is subject to a huge speculative literature. Okay? It might come, for example, from polarization of ultraviolet light hitting meteorites before they hit Earth, polarization caused by magnetic effects from the star. Okay? But that's all speculation. Okay, so what we did is we looked at the origin of transfer RNAs, and we came up with our own set, which are the ones in bold. Like Alan. Now let's see. Oh, this is showing for different meteorites: Asuka, Murchison, Paris, and total Okay, this is the fraction of amino acids from these meteorites and their enantiometric excesses, which are K values. Okay?
Okay.
So alanine definitely shows up in this list of observations of meteorites. It's been a while since I looked at this figure, so I'm a little slower. Okay, now, what we next looked at is the thickness of membranes of the first vesicles. And here's the... Okay. One of the problems is that meteorites might be contaminated, some of them. So we indicated which observations are speculated might be contaminated. Usually they go inside a meteorite, take some fresh stuff from the inside. But some meteorites are very porous, so living things could crawl in them on Earth. So that may account for some of the observations of microorganisms in meteorites. You put a motile organism in a meteorite, you don't control for humidity, and after 100 years, voila, you can crawl all the way inside. Okay?
Yeah.
So I've written papers on that stuff. But anyway, let's see. We got here. Okay, these are chainlets of molecules that are amphiphile in different meteorites. Okay? And you can see they're fairly short. They're 8 carbons, whereas our current membranes are about 16. Okay?
Yeah.
Okay, so this is where the hypothesis came from that current membranes had to be formed by some process to make them thicker. Okay?
All right.
The initial ones might fit thin. Okay?
All right.
Okay, so then I checked the carbon-carbon distance literature for chains. The chains are zigzags, so these numbers are a little bit weird. But a couple of estimates, two different estimates, they're reasonably close. Okay?
Okay.
Okay, then I checked the thickness of modern membranes. And these are the lengths, and these are the spacings between the carbon atoms they report. So they're not too far off.
Right.
Okay. Okay. Again, it's the distance along the chain of a zigzag of carbon atoms. Okay.
All right.
Now there's this question about whether or not they're bilayers or they have U-shaped bipolar molecules. I haven't found any literature that settles this question for archaea, which have bipolar. Current existing archaea all have bipolar membranes, which means that they consist of a long molecule which has a charge at both ends. But it's not known whether the two charges at the two ends are on opposite sides of the membrane or on an individual side of the membrane, in which case the molecule would bend and it would be U-shaped. Okay?
Right.
So that's one of the unsettled questions.
Okay.
Okay?
Yeah.
It's a little hard to see a molecule and see if it's bent or not, I suppose. Okay, now what do we got here? Estimates for thickness of the hydrophobic portions. Okay, we got those formulas. Extent thicknesses. Okay, so here are reported thicknesses of existing membranes. And you can see that they're about twice as big as a C8. C8 means 8 carbon atoms in a row, plus a charge group at the end. Okay, so they're about double.
Right.
Okay.
Yeah.
Okay, so here's the hypothesis. We assume that there's a feedback process going on of some sort, and it's been, I think I've got an illustration of it. Okay, here's a distribution of linear dicarboxylic acids, and you can see they peak around C8, C9, and our membranes would require C16. Okay, so the idea that they're around C8 is plausible when they for the first vesicles. Okay.
Okay.
Now, what do we got here? Estimates of how many. Oh, okay. These are estimates of how many. If you look at a membrane-bound peptide, how many? This is a question. How many amino acids are there from one side of the membrane to the other for a peptide that's stuck in the membrane? And these are the numbers. Got 16 to 25. Okay.
Okay.
Okay. And the things in brackets are all references. Okay, so
can I just have words? Barry here just saying hello for 5 minutes. I can't be too long. But you've just caught my attention because I was a peptide chemist with several papers on transmembrane peptides.
Oh, yeah.
Are these, I came in a bit late, are these alpha helical?
the presumption is that that there are alpha coils and beta coils. I only concentrate on alpha coils, and they're presumably helical and they're presumably hydrophobic.
So it's 1.5 angstroms per turn, I suppose, yeah.
Yeah, that's what we're calculating roughly there. Okay, so here's a simple model, and that is if you look at the growth of the membrane, it saturates at some value, which we assume is the existing membranes, which fits with that thermal curve I showed you originally. Okay?
Yes.
Okay, now let's see. I have a good picture of this. Okay, so here's the, oops, too fast. Where'd it go? There it is. Okay, so here's the basic idea. There's a process going on where peptides are growing, are forming, amino acids are polymerizing, and occasionally you get a hydrophobic one, a sequence of hydrophobic ones, which will then insert themselves in the membrane. So the idea is, at the top left, is that these peptides, since they keep growing, they can be thicker than the current membrane. Okay?
Yeah. Okay.
And as a result, there is a selection for membrane molecules that are longer, because there's a force pulling up, and therefore longer ones can be attached to the membrane peptides. And so you get a positive feedback mechanism here, where the longer the peptide goes, the thicker the membrane becomes. And the thicker the membrane becomes, it can accommodate thicker peptides. Okay, so that's the positive feedback that's postulated here. Here you can see this one's sticking out. I would bring in. Now we calculate in a separate paper that the number of day-night cycles available for these polymerizations of peptides are about 10 to the 11th. And that's assuming that when the moon formed, the Earth spun up because it was hit sideways by a rogue planet called Theia. And the original spin of the Earth may have been as short as 2 hours to go a full cycle day-night. So the Earth may have started out with a day-night cycle of 2 hours, and as the moon drifted further and further, it came down to current 24 hours. And there's lots of evidence, there's paleontological evidence that the day was shorter. I think the paleontology evidence goes down to 18 hours. Okay, but the calculation from the formation of the Earth is somewhere between 2 and 6 hours for a day-night cycle. So the assumption is each day-night cycle you might have been able to alternate the temperature, the UV exposure, etc., and this might have led to the formation of and growth of the peptides. So the energy source, so to speak, is the spin of the Earth. Okay, so the idea that the peptides keep growing is plausible.
Yeah.
Okay, I'm going to skip over this. Not going into what the RNA world is. There's a weird speculation that because some RNAs are self-reproducing, life started out with RNA. In this model, life starts out with vesicles. Okay, it's the most popular idea, and I think it's probably wrong. Okay, but go back in time and tell.
Yeah.
Bring your centrifuge with you.
Yeah.
Okay, now this is. Let's see, what are we doing here? Okay, here peptides going to membranes, and this is one way they could get across the membrane. But in any case, what I'm doing here is showing that if you get some from outside, a dead protocell, not that they're alive, but could absorb peptides, which would then go into the membrane and then could go into the vesicle. Okay.
Right.
So, Bradley, you see that the model that we're working on right now is a gross simplification of this.
Right. Yeah.
Okay. Okay. So I think that's it. That's basically the idea.
Okay.
Yeah, a feedback mechanism that seems to saturate might have occurred near 100 degrees, but the membrane fluidity goes up, and maybe other properties go up and saturate around 100 degrees. And this may be ultimately what sets the limit to the temperature for life, which I think is now the only I think there's one organism that can grow at 112 centigrade.
Okay. Yeah.
Okay. So it's about right.
Okay.
So it might have been hot, and it might have involved this feedback, and this also then gives you. Oh, I should say one thing. If you look at human beings, 1/3 of our proteins are membrane bound. So it could be a very primitive structure. Despite all that RNA and making everything, it's still 1/3 of what it makes sticks in the membrane. Okay?
Okay.
Now, there's also beta strands which can do this, but I ignored them because they're tilted, so you have to take into account their angle also. Okay?
Okay.
So this theory in this paper is obviously just a crude beginning.
Yeah.
Okay?
Yeah.
Okay. Okay, in summary, following events. Bombardment of the Earth by meteorites containing amino acids, and oops, moving too fast here. Oh, I'll go back. Okay. Stop.
You just pinged an old paper of mine.
Oh,
Is there any dependence on secondary structure prediction in this? Are you looking at specific sequences at all? Or are you just looking
Oh, no, we're assuming random sequences.
That's what I got, yeah.
Yeah, they're random, except that there's a selection for hydrophobic sequences.
Yeah.
And in fact,
Yeah.
I'm looking for somebody who's good at molecular dynamics to try to calculate the hydrophobicity of a random LD mixed peptide.
I don't run dynamics these days. I
Yeah.
was always molecular mechanics as a slight preference anyway, but there's plenty of programs, there's plenty of guys that can run dynamics. Right?
Oh yeah, they're there, but they're not interested in the origin of life.
No.
I know.
You could.
Yeah.
There are ways to calculate the end-to-end distance from assuming independence under random coil conditions. Actually.
Yeah, but see, nobody does mixed LD peptides. They're all calculating L peptides.
Well, later I wasn't, but the sort of thing that you were, yes, they were L.
I'm sorry, I missed that. What?
I said I've done a lot of work with D and L proteins, peptides later, but the sort of calculation you're thinking about, they weren't mixed. But it really flip the calculation, because if you're treating them as independent, you're just going to mirror image, right? So.
Well, no. That's not. No, I'm saying the individual peptide is a mix of L and D.
Yeah, I understand. Oh, so you're not saying it's necessarily a random mix within the peptide.
Yes, I am.
Oh, well, yeah, okay. Then they're saying you assume random coil independence of the amino
Yes,
acid.
and then you get selection towards those that are hydrophobic because they fit in the membrane. And then you might go another step, and there might be some sort of selection towards L molecules, given the L bias in the meteorites, or given some sort of structural thing with the membrane. There's a possibility that there's also a solution to the chirality problem here. For those of you who don't know, the chirality problem is why do we have mostly L peptides, L DNA, I think, and D sugars. Okay, there's a huge literature speculating on why this might have occurred. But here, I think we're going to have to get to molecular dynamics to figure this out. Okay, so Bradley, you got now the thing that we've added in the paper we're working on is that if we go, let's see if I can find that curve. If we go back to the first curve, I'll find it. This one. Yeah, this one.
Yeah.
Okay. Suppose instead of temperature, the vertical structure, the y-axis is permeability.
Okay.
Okay. So over time, the permeability decreases. It starts at 1 at the bottom, goes to 0 at the top.
All right.
Okay. But as you do this, you're getting more and more peptides, and you might end up with some peptides that cause the membrane to leak through the peptides or through structural alteration. of the membrane due to the peptides.
Right.
Okay, so there's a possibility of a mechanism setting in to get what are called ion channels, which are special proteins that allows things selectively to come through past the membrane. Okay, but our simulation does not include that. We just go to the point where it closes, where the membrane itself closes up entirely.
Right. Right. With nightfall, actually.
So,
yeah. Or the one we're working on. Or the one we're working on, it's nightfall where things get trapped in there, and then daytime it's permeable.
Well, but during the daytime we'd have lower permeability of the membrane.
Right. Right.
Okay.
Yeah.
Now you brought up an interesting excuse me, an interesting point, and that is that the membrane might be asymmetric, because after all, you got these peptides on one side and you got water on the other side. So there's a possibility that the R in and R out could also change the ratio of the two. Even though both are going to zero, the ratio might change. Okay, yeah. So I guess I'd like to keep that in as a feature of the simulation, but for now, just assume that R in is equal to R out all the time and see what happens.
Right. Yeah. Yeah.
Okay. But this is the basic idea, that this is not just a plot of temperature, but it's also a plot of permeability as the membranes get thicker. Except that it's 0 on the top and 1 on the bottom.
Right. So you get this log linear curve, but it's just going to 0.
No, it's not log linear. It's not log linear. It's linear.
Oh, okay.
N in this case is the thickness, basically.
Oh, okay.
Measured in carbon atoms.
Oh, okay.
Okay. Okay, that's it.
All right.
Okay, what did you? Angiogenesis of transmembrane domain.
Where are you reading?
Yeah.
Oh, just the top. What are you calling
abiogenesis. You're reading the wrong word.
Oh, okay. Biogenesis.
Abiogenesis means that before there was life, everything was dead.
What is the title of your paper?
Oh, I forgot.
No, we'll read that with interest.
Let's see, what's the title? Gotta get up. There it is. How to make a transmembrane domain. The domain refers to a portion of a protein molecule at the origin of life. Okay? So this is one possible mechanism for making proteins, by having this positive feedback mechanism that selects for the portion of the peptide which is in the membrane has to be hydrophobic. Okay, so it's a selection without the
Okay.
Okay. And this could be made more complicated. If we actually knew the distribution of molecules, you could also say, okay, how do you make a membrane with a bunch of amphiphiles that are of different lengths? And does that cause selection for those that are the same length as the current thickness of the membrane? Okay, this model could be made much more complicated.
Yeah, I think the transmembrane proteins that allow ions to flow in and out are extremely important to life.
Oh yeah, those are the channels. And obviously, if the membrane's closing down by becoming impermeable, at some point you've got to have domains. You have to have ion channels. Okay?
Yeah.
And the ion channels were also essential for creating a charge difference between the inside and the outside.
Yeah.
Okay.
Yeah.
And then you have the whole problem of how do you get chemi-osmosis going? Okay. Okay. So this does not solve everything.
Okay. Looks like sort of just start.
Yeah, it's sort of a start, and if it's something you could actually experiment with, I suppose. But
yeah.
There's nothing in here that's that is not something couldn't be brought into a laboratory. Okay.
Yeah.
Of course, if you need 10th to 11th day-night cycles, you're going to have multiple generations of people, but we don't know if this happens in a couple of days or if it takes 10 to 11th days.
Oh, well, I know the physics conference, APS March meeting, they had someone do a cycle of DNA and RNA to see what combination survived, and it turns out to be the one we've got. So I think that was interesting.
How many cycles did they simulate?
Thousands of years
Thousands of years of what?
of survival of the form, the molecules.
Okay.
I'll
You have to be very
send it to you.
you have to be very careful in this business. If you know what you want, you have to make sure that the goal point is not the measure of what the thing is going towards.
I thought that they were very careful about
Okay.
Yeah, I did. So
Yeah. So it's hard. This gives a positive feedback mechanism and a hint of a saturation with that curve on the temperature. So it's only a factor of 2 also. So, you know.
Right.
So I'm just taking that factor of 2 and making a big story out of it.
Right. So where do the vesicles?
Contrary to the way
Go ahead.
Yeah, it's contrary to the way it was brought up. If the theory is good within a factor of 2, it's probably okay. So here I'm nitpicking over a factor of 2.
So where do the vesicles come from? I'm just curious.
Oh, they come from amphiphiles that are found in meteorites.
Oh, okay. So that's the okay.
Where they came from, I don't know.
Yeah.
Okay.
Yeah.
So you know, there's lots of evidence for amphiphiles and meteorites. There's only one paper where somebody actually bothered to make vesicles from them, and they did that in a very qualitative fashion. And I was told that that time when they did it, the chemistry of the meteorite was such as they couldn't tell you what molecule they were dealing with.
Yeah.
They just know that they seem to make vesicles. They saw them under the microscope, and therefore they must be amphiphiles.
Yeah.
Right?
Yeah.
Yeah, I'm sorry. The evidence for this field is very sparse.
Yeah. Yeah.
Okay.
All right.
And what we really need is a time machine. But
Well, yeah, that's not gonna happen.
of course, we probably contaminated everything.
Yeah. Well, that was great. Thank you.
Okay. So that gives you a little better background on what we're trying to do.
Okay. Yeah, that's great.
Stop sharing.
Okay.
Okay.
I think I wouldn't claim to be the first to do what you're doing. It looks pretty original and ingenious to me, but there's some surrounding areas for Purell I got in my old textbook that I wrote there. So there's a chapter on random coil peptides and dimensions and things like that.
Oh, gee. Here, let me.
You can get that for 10 cents from Amazon. It's not a problem.
That's okay.
What's the title?
Introduction to Proteins and Protein Engineering.
Oh, okay.
Here,
If you ever heard of the Gore method for secondary structure prediction on the R in that?
Okay, here. Let me put this on the screen. Do me a favor, send me that. Can you read my email address?
Not yet.
Yeah, try to magnify it or something. I don't know if I can magnify it.
I don't think it's in the chat right now.
It's not coming through on the chat.
Okay, I'll put it in the chat. Okay,
Yeah.
I'm not connected fully to you guys yet by email.
DevoWorm chat. Okay, open chat.
I can. I'll take a screenshot of it and then I'll
Okay, here it's in the Jitsi chat.
just make sure I've captured that.
You can just copy it. Yeah, just copy it.
If you hold it up, I can do print screen.
See,
Okay, it's in the chat, so you can copy it on the chat. Yeah, I'd appreciate that because there's, you know, yeah, the structure of mixed LD peptides. I really couldn't find much literature on it.
Well, my later area, which was especially active in the first month of the COVID, is peptide-mimetic drugs, which are partially made of D.
I'm aware.
Glucolysis, yeah.
I'm aware that people do that occasionally, but there's no systematic study. I'd like to know if a random LD peptide is hydrophobic or not. And if you have a bunch of them, can you select from the ones that are hydrophobic?
Well, it depends on the composition. You start with the amino acid soup, of course, which depends on that.
Well, of course, but let's start from racemic mixture and see if there's a selection process. You can always start from an antimetric excess.
Yeah, I think those early days. Yeah. The only thing I make of what was very popular at the time was that these things did not come from other planets or meteorites, but they just happened to form at the vent temperature around the rim of volcanoes.
Oh,
so I was a little biased there, but that was the
interesting. So anyway, any questions about Bradly? Is it obvious now?
Yeah, I see. Yeah, I see how.
So we're summarizing this by speculating that the membrane permeability starts at 1, which would correspond to the 8 carbon atoms, and goes down to 0 at 60.
Right.
Okay. By which point something had better give, because at that point, which is we're not simulating, you've got to have some sort of ion channels, otherwise things that never gets alive.
Right.
Yeah. Okay.
Yeah.
Okay. So I don't know how ion channels form, but I suspect that the disruption of the membrane structure as a two-dimensional fluid, the disruption of it by the peptides that are sticking in the membrane might be a source of the initial channels. You know, but that's going to require very detailed molecular dynamic simulations.
Right.
Okay, which is way beyond me.
well, people don't think on that. They just buy a program and run it these days, much to my chagrin.
You have to learn how to run it.
Yeah, right, that's the hard part.
You don't want me behind the wheel of a car.
I don't even understand Windows, so I'm not going to help come with those.
So that's our meeting for today. I look forward to seeing you next week.